Hi, and welcome to the Biology 141 semester review. We decided to make this video to give you an opportunity to see the course material actually applied to a disease, chronic myelogenous leukemia, or CML. Now, this is not going to be a review on the transcription translation mechanics. We've done that previously in a lecture. The nice thing about these lectures is you can go back and play it again. What this is a chance for you to see this course material applied. So if you actually look at these images below, you can see how the disease is affecting blood formation. The slide on the bottom is a blood smear from a normal individual. Notice how you have a majority of red blood cells with some platelets interspersed. And then there's the white blood cells. These guys are stained a little purple. These are neutrophils. Now the ratio is important because this individual will have a normal physiology. The slide on the top, notice how you have a tremendous amount of these darkly stained white blood cells. This is a hallmark of leukemia. So let me start by saying that cancer is a genetic disease. Chronic myelogenous leukemia, CML, is no different. It's a change in a gene expression, and this change in a gene expression and the gene produced leads to the phenotype. So let's get started in talking about applying this course material to CML. Okay, so here's the key topics we're going to be covering in this review. And notice that it's heavily weighted toward gene regulation. We're going to be covering transcription factors, what they are and how they work. We're going to be talking about coordinated control of gene expression and signal transduction cascades. We're going to be hitting on gene regulation and epigenetics. We're going to briefly mention transcription and translation mechanics. And then we're going to talk about how post-translational modifications play a role in the process of CML as well. Now, cancer is a disease of the metabolism and messing up homeostasis, and how the cancer itself is caused by mutations. Finally, in part 2, we're going to be talking about CML resistance to Gleevec, and that's going to directly relate to antibiotic resistance that we saw in the bacteria. So the key topics indirectly related are going to be prokaryotic versus eukaryotic gene structure and mRNA processing. So a very important topic in cell biology and developmental biology is the concept of cell differentiation or cell specialization. And essentially what that means are cells start out as a progenitor cell where they're not very specialized. They essentially have many paths they can choose. And then what happens is as they grow and divide, they become more specialized. So they become more highly fine-tuned for a job. As they do this, they lose the ability to divide, but then they get better at doing their job. Now, one of the hallmarks of cancer is that these cells screw this up. They regress and they go from a more specialized cells into a progenitor cell that then essentially takes over an organ or tissue. So intuitively you have an understanding of cell differentiation. The sperm and the egg meets and you get that first little zygote, and by definition that one cell is going to be able to produce every cell, every tissue, every organ in the body. So what's happening is those cells need to turn on or off genes, and as they do this they become specialized, they become differentiated. So that's how they're able to produce all the entire skin tissues, fibroblasts, intestinal cells, all the specialized cells in our bodies. So how do cells differentiate? What is this mechanism? I keep showing myself off from my college days. But how does that zygote form specialized small intestinal cells or muscle cells? Well, it has to do with the concept that you guys are familiar with, heterochromatin, euchromatin. So remember in gene regulation, we have essentially two types of DNA residing in our nucleus. These darkly stained regions are heterochromatin, and these lighter stained regions are euchromatin. Now, this relates to the accessibility of DNA within those structures. So for instance, take your Christmas ornaments. In the middle of summer, you have no use for those, they're not going to be out. So what are you going to do? You're going to pack them up and put them in your attic, and they're all going to be tightly contained and wrapped up and inaccessible. Well, that's equivalent to what happens to genes in your heterochromatin. So a muscle cell has no need for the small intestinal cells. So what's it going to do? Well, it's going to package them up in a nice, neat container, and we call this container heterochromatin. So your Christmas ornaments in August would be the heterochromatin, and your muscle cells would be doing that to all the genes that they don't need at the time. Now what happens come Christmas time, or actually Halloween, with the stores nowadays letting all their Christmas stuff out around Halloween? Well, those ornaments need to be opened up and used. That's the equivalent to the euchromatin we see in our cells. The euchromatin is DNA that's opened up accessible, the transcription factors can get in there and transcribe that gene to make messenger RNA and protein. So when we think about cell differentiation, that's actually what's happening to the cells. Your muscle cells will have all the genes that they want to use to become muscle cells, to be muscle cells, opened up, being transcribed and translated, and making proteins from those. Your muscle cells are going to be turning off your intestinal cells, your intestinal genes, and those genes will be packaged up into the heterochromatin. So in this way, this is one of the hallmarks of differentiation. As a cell becomes more highly specialized, they're going to package up certain genes and open up other genes that they're going to be using. So again, running with my differentiation Christmas analogy a little further. By the way, how cool are those Christmas ornaments? So pretend like it's Christmas time and it comes for you guys to go home for the semester. So you're here in DeLand, and one person may live in California and one person may live in Michigan. You guys may all both start out hopping on I-95 heading north. Well, that's what happens with your progenitor cells. They start out as being completely able to produce any tissue in the cell. As they progress through development, they start to make decisions. So at some point in northern Florida, the person going to Michigan is going to make a right-hand turn and head up I-95, and the person going to California is going to make a left on the I-10. So right there, you guys have made a choice, a decision, and the analogy for a cell is that cells are going to start turning on certain genes and turning off certain genes. And as the cell progresses, as it gets closer to its destination in California, more and more genes are going to be silenced and other genes are going to be turned on. The person heading to Michigan is going to have a completely different set of developmental choices that have occurred. These modifications, the heterochromatin, euchromatin packaging, is going to be different between these two cells. The cell de-differentiates, it becomes unspecialized, and essentially has to drive the car backwards. It has to rewind its fate on the map. So one of the more interesting questions that arises that often gets asked about cancer is, so you have a bunch of cells growing out of control and too fast. So is having too many liver cells that bad? So why does cancer actually make you sick and possibly die? Well, the answer has to do back with metabolism. The metabolic processes in cancer cells and in individuals that are experiencing cancer are actually disrupted. So there's two main ways. The first one is the function of the tissue can actually be disrupted. And the second way is homeostasis of the organism as a whole can be disrupted. So for the function, you can see here the top slide has a normal lung cell. So you can see all these nice big open sacs. So when the person inhales, the air can get in there and you can have exchange of oxygen and carbon dioxide as the person respires. Well, the slide on the bottom is somebody that has cancer. It's the same location, or could be the same location in the lung, and notice how the tissue looks markedly different. The person on the bottom is going to have serious issues when it comes time to exchange carbon dioxide and oxygen. Now, our organs and tissues, they tend to be pretty tough, pretty hardy, so you wouldn't necessarily notice a small cancer, but what happens is by the time you exhibit symptoms, the function of the lung could be severely compromised. The middle picture is kind of gross, but what you're looking at there is a colonoscopy of someone. And in there you can actually see a mass of cells, and those cells are disrupting the normal function of digestion. So this person eats, and the food may literally get stuck on top of that mass. It can no longer pass through that mass. So again, the function of the digestive system is being inhibited by these cancer cells that are just growing, right? So they're disrupting the function. The other way is a little more insidious. Homeostasis can be messed up. So on the top, you can see someone that has a bone cancer. And notice how that tumor, that cancer cells right there, are growing and they're not really formed very well. But what they're doing is essentially acting as a nutrient sink. Calcium is being sucked out of the body, sucked out of the blood, to allow these cells to grow out of control. And this can severely disrupt calcium levels. Remember, calcium plays an important structural role, so this person may have osteoporosis developing, they may have weakened bone in other areas. It plays a role in signaling, so their muscle contractions could be messed up. You can end up with heart issues. And also, calcium can act as a coenzyme, so their enzyme activity could be messed up. So this simple tumor, as it improperly takes calcium from the blood, can actually cause a whole series, a whole cascade of effects. So below the x-ray of the bone cancer, you can see someone has had their liver removed, and all these nasty white masses are actually tumor cells that have arisen inside the liver itself. The liver is actually an amazing organ. It has a tremendous regenerative ability. which is a good thing for those Saturday nights or homecoming nights when you guys do a little extra partying. But this has been recognized for centuries. And Rubens actually did a painting. It's in the Philadelphia Museum of Art called Prometheus Bound. The legend of that is Prometheus gave fire to man, and as a consequence, the gods chained him to a rock, and every day an eagle came and ate his liver. So even ancient Greeks, in their stories, they realized that the liver has a regenerative capability because the liver regrew in Prometheus, and sure enough, the next day the eagle came and had another meal. So the liver also plays a significant role in maintaining homeostasis. It secretes a tremendous amount of proteins to regulate blood plasma levels. It produces cholesterol. It releases amino acids into the blood, so a role in protein synthesis. And it also produces bile. So the liver itself is very important for a person to have fully functional. And as you get these growths inside there, what they can do is actually cause an increase in release of certain hormones. So the person may not even know that they have a cancer because the liver itself can get bigger. You know, it's a soft organ. It can get bigger without being noticed. Those tumor cells can actually be secreting hormones that are really messing up the person's homeostasis. And those hormones can actually cause someone to die. Okay, so let's get back to leukemia and talk about CML. Now, remember you saw in the differentiation picture that hematopoietic stem cell gives rise to the red blood cells, the white blood cells, the platelets. And what happens in leukemia is that the hematopoietic stem cell is not necessarily off, but the progenitor cells can be overproducing themselves. So this is a cancer of the blood and bone marrow. You end up with an increased number of abnormally functioning or white blood cells that are not functioning. So the cell cycle has been short-circuited in all these cells, as we saw in that first picture with all of those blue darkly stained cells. These cells also tend to de-differentiate. So the two of those things combined can lead to some pathology. The bone marrow only has so much space. Inside the bone is a hard structure, and if the bone marrow becomes filled up with these leukemia cells growing out of control, it can actually interfere with normal red blood cell development and platelet development. Now, this can lead to fatigue, excess bleeding, increased likelihood of infection, and actually some leukemias are horribly painful. Your bones are almost breaking from the inside. It's really, really sad condition. So all this taken together can give you an idea on how important it is for homeostasis and metabolism to be maintained within cells. And these cancer cells, these leukemia cells, are disrupting it. They're short-circuiting, they're growing out of control, they're taking up space that they shouldn't be, they're taking up nutrients that they shouldn't be, and they're not functioning the way that they should. So all this can lead to the pathology.